It's quite an interesting looking landscape, seascape.
Tilting up. Oh, I see some other bubbles.
So people think of bubbles as just sort of these nice little idealized things you see in your soda glass, but the reality is that in the ocean, the bubbles become what we call dirty very quickly. So they have all those little tiny particles that sit on top of the bubbles and actually change the dynamics of how the bubbles interact with the seawater, both the physics and the chemistry, pretty substantially. So there's a whole different theory about how dirty bubbles behave versus how clean bubbles behave, and we just don't think we ever really have clean bubbles in the ocean.
Huh.
Wanted to take another look to see if these were more pieces of bivalve, which might indicate there are bivalves living down here below the surface and not just being transported in. And maybe we see one of the reasons they're all chopped up right here.
Rotating. Okay.
So maybe these are some of your chemosynthetic organisms?
Yeah, there definitely is a bivalve that's been collected. I think that we collected some in the Mid-Atlantic Canyons project.
ROV Nav Bridge.
All right.
Just wanted to give you an update on our current.
Let's keep moving. Rotating up.
At 0.9.
Be nice to get a whole shell as a sample.
Hopefully we're going to come across a actual live patch of them. That would be great.
Yeah.
But I guess if we don't, we can pick up a shell.
Nice fish, everybody. It's a rather large hole next to that pout.
I don't know how far that is coming.
We're getting close to our waypoint 3. There's a McTophid, which is a midwater fish, the lanternfish.
Look at his teeth.
I'm drifting back.
With one of his predators lurking in the background there.
Yeah, nothing to see here. Just keep moving along.
Sorry. All right, let's let it go. What about this? Is this? Oh no. Okay.
I'll look inside of one of the holes.
Another siphonophore.
Let's.
And we're now at 468 meters. We're seeing some more burrows here. We haven't seen any of those in a while.
Just continue to fly over it.
Yeah, that's max. Oh no, I have a little bit more.
I don't think we have to stop, just continue to flutter over it.
Okay. Coming up. Tilting up.
Tina pointed out that the organism with the black dots looked more like a salp.
Oh, the chains of...
Mm-hmm.
Oh, really? Oh, there's some bubbling.
Bubble came in a big dot in the, right next to the lasers, too.
Okay, hold on, 'cause I buried myself. Hold right there, hold right there. Hold. No, I think we're fine.
This looks a little more bumpy.
Mm-hmm. Yeah, I was looking around to see if there might be any bivalves here living.
Okay, I don't see any more bubble action. Let's just take a look at this closely though. Come in.
There's that little red worm again. Or whatever it is.
Yeah, whatever it is.
There's another one, so. One at the top of the screen also.
Yeah.
Can come up. Pushing out. Tilting up.
And more eel pouts. Wow. Oh yeah, so that one right there was probably a salp. And those are
something large off to the right in the distance.
All right, Nav, you ready for a Niskin?
What's that off in the right?
All right, watch lead. This is co-pilot. I'm going to take a Niskin here.
Roger that. Thank you.
Anemone.
Do you want to wait until we're directly over waypoint 3?
Sure, that'd be great.
Okay, come in. Or hold right there.
I'm assuming this is one of the tube anemones.
Wow.
It's got a, does it have a salp chain?
Very prominent compared to the others we've seen.
Come in. Hold.
You can see the long extensions of each of the tentacles.
I wonder if he's caught a salp, and that's what the dark brown circles are.
That's what it looks like.
Am I drifting? I don't think I am.
Look at your toes. I'll tell you what you're doing.
Squid's digging at the base. I wonder what he's after.
All right, if we want to, is the ship still moving or are we stationary? I think we're stationary.
I think we're moving right now.
No, we're stopped now.
Okay, copy.
Is that a tiny squid?
Yeah.
On the bottom?
Can we put the lasers on when you...
Wow, that
zoom
guy's
out?
tiny.
Bridge. Move complete.
That's just a big anemone, too.
Yeah. Wow, that is a big anemone. Right there. Uh-oh.
Oh.
I wonder if he's stuck in the sticky
he might
tentacles.
be stuck and he's trying to get out.
Attached.
No.
Got away.
Seems to be okay.
It's amazing they're translucent and you can see
ROV
right
nav
through to
bridge.
their stomachs.
If now's a good time, I would like to adjust our heading about 5 to 10 degrees to starboard. Okay, thank you.
So is this the helix then?
I don't think so. I think it might be that other... I have a hard enough time telling them apart when they're large.
Video's clear.
Okay, let's come up.
And if you want to drive to waypoint 3, we can take that Niskin. So, to your port.
Where is it? Oh, okay.
It's right underneath Sirius.
Wow.
You guys want to collect the Niskin at the waypoint or
Yes,
above one of
they
these
want it-
mats?
They want it at the waypoint.
It's very impressive.
So,
The starboard.
oh,
Push forward a little bit.
That's
Hey,
really.
Mike Becky here again.
Hi, Mike.
Hi, Mike.
Basically center up in Sirius.
Hey, yeah, I think that was a brachiotesis that you got a close-up on that looked like it was trying to dig its way through the center of the Earth.
Uh-huh.
I think what's going on there is that it was trying to get away from the light, and the typical way for a vertical migrator to get away from light is to swim downward. But unfortunately, there's a bottom in the way here, so they keep running into it. But you have seen ilex too. I've seen several ilex sitting on the bottom, which they typically do. I guess I've never seen brachiotesis resting on the bottom.
So, Mike, the ones that are propped up on their arms are the ilex?
Yeah.
Okay.
They look like they're resting on their elbows.
Yeah, yeah. Okay. But most of the ones that we're seeing are the other
reason
type.
they do that is to keep their funnel up out of the mud so that they can breathe okay.
Another fish in a hole to the right.
And I did have a question. You did a close-up on a fish that someone referred to as a toefit, I think. It was a
thing we're
it
gonna.
was a viperfish. It was a really nice close-up of a viperfish.
Oh, that came up into the view right in front of us?
You guys know that.
Yeah.
Okay, that was my bad.
It was some time ago.
I need to get better with my midwater fish identification. I guess one set of
that's the one
midwater
with the
transects
really
is
long
not
teeth.
enough.
It's got teeth that poke up through the bottom teeth, poke up through the top of the head when its mouth is closed. And when it's swallowing something, it can unhinge its jaw to get a large prey down.
All right, we caught that.
Oh my gosh.
There's something
We
going
just
on
saw
down there.
the burrowing fish. Mike, I know you're a little delayed or you can't even see the video, but we just saw a great
Did
little...
the fish come out and grab the squid, or the squid just buried?
No,
I don't know. We lost both the fish and the squid in that one.
All right.
We'll look at the replay.
I don't think that squid had much of
Okay.
a chance to take out
Well, I'm going to sign off now.
Okay, thanks very much, Mike.
Yeah, it happened so quick. I wondered
bye.
if the squid just freaked the fish out.
Well, I think the squid went down with him.
Yeah.
Oh, it did eat it. That was so cool. We'll have to make sure that
Okay,
we
let's
get
move
this
out. It's a
video
little
up
bit too
online
cloudy.
at some point, but the replay of what just happened,
Yep.
the fish in the
And
burrow
at this
opens
point,
his mouth and sucks in the whole
so
squid.
Now that we've
It was
now that
crazy.
we've reached waypoint 3, we'd like to go.
We've got it on stream 3 right now.
waypoint 4 and
The
start
replay is on stream 3.
Yeah,
What
check
is,
out Stream
bearing
3.
to
It's
waypoint
way
4?
worth it.
It'll be pretty much
It is so
west.
neat.
West.
Can you just do a slow
Yeah, you can see
replay again?
over
Okay.
here,
We're going to back it
it's
up
waypoint 4, so
if
we'll
you're
stay at
watching
this deck and
stream
just kinda
3.
ride this contour.
Okay.
Well, we know one thing that fish eats.
Looks like the squid was probing at him.
Either that or he just got unlucky.
We're in super
And
slow.
then, how do you want to travel?
Yeah, I think he got unlucky. He hit the top of the fish's head.
Yep, not a good place to be.
Oops.
What was that, Nick?
How do you want to travel? Do you want to look west or cut it at an angle behind?
It's not steep. I mean, we could just head in that direction.
That was great.
Copy.
Thank you, Art Howard, for that clip.
Do you want to do 30-meter hops at decimal
And that
one?
reminds
Sure.
me, we haven't had introductions on the front row.
We'll put one in.
Pilot at all? Would this be a good time?
Yeah, I think so. Hello everybody, this is your pilot speaking. My name is Fernando Aragon. We are all part of the Global Foundation for Ocean Exploration, and sitting to my left
is navigator position Sean Kennison.
And in the co-pilot position is Josh Carlson.
And in the far right is Roland Bryan in video. And in the back you see Art Howard on the left of the screen. And then our two esteemed scientists.
Hi, I'm Leslie Sauter from the College of Charleston. Hello, South Carolina Aquarium.
And I'm Cheryl Morrison from the U.S. Geological Survey. Hello, Aquarium. And everybody else. Thanks for joining us on this Saturday morning.
And also hello to Zach Prue, who is in the visualizations lab at the College of Charleston. He's hosting visitors as well today. So if you are in Charleston, either go visit the South Carolina Aquarium or the Science Building at the 202 Calhoun Street. You'll get to see large screen representation of our live stream, but also some commentary. You can ask questions.
Hold on 1 second.
And Roland will be swapping out with Bob. Bob will be taking over video.
Copy.
For those who haven't seen a lot of seep sites before, this is
Are you switching before
very
this or after this?
expensive bacterial mat that we're
Okay,
seeing
then
here.
let's come in.
Amanda Demopoulos, who dove here last year with another vehicle, so these are some of the largest bacterial mats she's ever seen, and she certainly has surveyed a lot of seep sites over the years.
Yeah, that's very good perspective, Carolyn.
Yeah. Good to hear.
Come in as you wish.
They looked quite extensive to me, but I have not seen as many as Amanda has in her travels across the deep sea floor.
Stay with his
It
eye.
looks like either a well-fed Limonema or a pregnant one. What do you think?
Well, with all the food around, maybe it's a recent meal.
I wonder if it's protecting a burrow, though.
Well, that could be.
Yeah, she's definitely circulating.
And Scott France said that it could be a parasitized fish. I can't see any parasites on the outside.
Okay, going medium.
Copy.
It's a great view of the hake.
Get lasers.
Yeah, and Scott said since the bulge is on both sides, the parasite idea might not be true.
So I've adjusted the lights on Sirius. We're down to one bank.
Okay.
Video's clear.
Let's come out and you can switch.
One of the barracudinas. Is that what you call them?
Mm-hmm.
Looks like a pencil sticking out of the mud.
That move was at 0.1 nav.
A rat tail to the right.
I put it to 0.2 since you got it a little bit.
All right.
I don't know if any of the fish biologists are online yet that might know whether Limonema hakes release eggs into pits. Because that would be interesting in that perspective. Maybe that's what that fish was going to do in the near future here.
Okay, Bob, when you're ready, we can come in. Hold right there.
Yep, and there's a hagfish head. And I didn't realize until this cruise
right.
that
Let's go in.
this is how they spend a lot of their time, just sitting mostly buried in the sediment with their head poking out and waiting for something to come by.
Like that?
Maybe something a little bigger and more fishy, but
wasn't listening to me.
All right.
He's trying to hide a little bit more now. And we've had the opportunity to see them burying into the sediment head first many times during this expedition, especially over the last few days.
Yeah, a lot of the black sediment brought to the surface here.
Partial ink video. Right there.
Oop.
That's the second one.
And
that
that's
hits
probably
that
why.
Yeah, we can come out.
So was Mike Vecchione saying that those squid, he doesn't usually see them on the bottom?
Yeah, that's what he said.
So it's interesting. They seem very attracted to this
My goodness.
area.
Look at all this bacterial mat.
Looks thick white.
Mm-hmm.
Do we have a lot on that move? Long way to go.
It's coming to its end right now.
Copy.
I'm going to turn to face you here, and then we can just keep moving to the west as the pilot is comfortable doing.
Copy that.
Okay, watch.
Got a hagfish right in the middle.
Let me know if you want any zooms.
He's living right in this bacterial mat, maybe.
I think that will be one of my landscape photos on our dive summary.
Yeah,
representing
amazing.
the substrate. That is
in the middle.
Very impressive.
You're ready, your lasers. Is that a squid?
Partially.
No, is that a black belly rosefish?
I think it is. Yeah.
It is.
Yeah.
Just sitting right on the mat.
I don't think we've seen those today.
With an eel pout.
And it looks like one of the cancer crabs up on the left.
Okay. Hold right there.
Yep, you got your left toe in.
An interesting scientific question here, of course, with these mats. Back 15, 20 years ago, we were calling them seafloor flux indicators, sort of flux of methane and or sulfide from below. And the question would be with the patchiness, is the patchiness mapping changes in the chemistry of what's down there, or is it just because of the way the mats grow and actually it's a very homogeneous area and the mats sort of start in one place and then grow in some direction, regardless of, as long as the right stuff is there for them to feed off of. The other thing to mention here is the juxtaposition of these really white mats and the black reduced sediment that some of the biota are stirring up. So obviously that just is a manifestation of the chemical processes happening in the sediments right below these mats.
Okay, ready. Whenever you're good.
So I wonder if before we go too much further, if we can pan to the right anymore and see whether these mats continue a little bit down slope, or whether we can see any more of that. Doesn't look like it, so maybe it's just up on this, the higher part of this ridge that we're following.
Cheryl, I don't know if that's the case, but often, of course, we see the gas coming out of the locally highest, sort of near the locally highest point, instead of sort of on the side of a slope.
Okay.
Well, and we saw that yesterday, at least the evidence for previous seeps. We didn't see any bubbling, but definitely on the high point.
We'll keep our eyes peeled for more bubbles as we make our way.
Pilots, are you ready for me to
towards the
put
top
another
of this?
move into west?
Yeah, I think so.
Copy that. I'll put another 30-meter move, decimal 1, if that works.
Copy.
Just keeps going and going.
Watch your left toe. Is that another sea star? Lower right.
This one?
Another black belly rosefish, and looks like another crab up here. Two black belly rosefish.
Bellies, yeah.
This one?
Yeah.
That's partial in video.
Yeah. Black belly rosefish.
Yeah. I think it's
and a
sea star.
mud
right
sea
here.
star.
Come in. Do a quick zoom.
He's good and buried.
Okay, let's come up. And
there's some bubbles right in the foreground.
Oh yeah, we've got bubbles. If we can just pan down, pilot, if there's time.
Does anybody know where those bubbles came from?
Probably right in front of you.
We scared them off. Try the larger hole right there.
We can come in video, but I don't see.
It's probably a intermittent stream.
Hold right there. Okay, we can come out. Okay, tilting up. And then let's just take a look at this and we'll keep going. Partial in. Is this just another crab? Yeah. Hold right there. Okay, let's come up.
So I'm facing 90 degrees from, or 70 degrees off of our direction of motion. You can stay up where you are, just to let you know.
Yeah, I'm trying to go
starboard.
Diagonal right here.
We can move Sirius a little more south. Just bump Sirius a little.
You want a southern bump?
Just a southern bump. Maybe on the next move, just angle it.
Yeah, sounds great.
Go ahead. Sure. Okay, let me know if I get too far out.
That's amazing.
Yeah. You got a little bit of a...
There's a seep right in front
Oh,
of
there's
you.
some bubbles.
Yep, just below.
2 of them.
They're all right below you.
I think.
Oh, there comes a really dark cloud.
Well, you certainly see how these bubbles are intermittent from place to place and through time.
Yeah, you can definitely push forward more if you want. I turned my heading more to face you. So watch your toes. And you're in the center.
I just feel like we're going to climb up and there will be this major plume coming out the top. That's what I'm hoping for.
And maybe a whole chemosynthetic community.
Ringed by mussels and clams.
Roger that. Thanks, Nav. We've just completed 300 meters.
Now, as far as you can see in the distance on that slope.
And if we could put the lasers back on for a minute, that would be helpful to get an idea of the extent of this. Thank you.
I'm seeing more current, I think.
Yeah, it's been intermittent.
These mats are so continuous that they're almost mounded relative to the surrounding substrate. They've been growing vertically.
is bubbles, right under the left laser.
The ship's moving. Navigator. Okay.
Moving south.
South?
I'm sorry, not south. Moving
Okay.
west.
All right.
Looks like we have a few more meters left in that, and then after that I can bump steer us a little south.
Let's come in video partially.
Another tiny Nizumia rat-tail fish.
Okay, we can let it go. Let's come on in video.
Class, we're going to pause a moment. They're going to turn heading.
It's a big fish.
Copy. I don't think I have enough tether to get out there, but...
That's okay. I think that's a southern hake, and we've seen quite a few of them the last couple days.
Okay.
Watch lead, stream 3
Right,
is currently down.
Video.
We are resetting.
Let's zoom out.
We're having another problem with stream 3, so we're working on it. Sorry about that. We're also having a ship
For you
move.
non-scientists or people who are sort of not the ones who sit on these calls all the time, you know, just the fact that there's not methane actively leaking out, and just because we don't see beautiful mussels and other organisms here doesn't mean this isn't a really dynamic and active area. These bacterial seeps depend on sulfide, and sulfide is actually a byproduct of the breakdown of methane in the sediment. So there are microbes in the sediments break down the methane, and one of the byproducts is sulfide, and so that's what the bacterial mats are sort of concentrated on when they sit on the seafloor. So this is still a very dynamic area, and sometimes you see some bubbles that do come out, but just because we don't see these sort of stereotypical gorgeous photogenic communities doesn't mean that nothing's going on below the sea surface here.
Come in, video.
Or the seafloor here, sorry.
That's a really good point, Carolyn. Thanks for that.
Yeah, and these bacterial mats look like they're thriving. They haven't been covered by sediment, so it would appear that maybe they've been
is there, and sulfide, of course, is toxic to a lot of organisms. Some organisms prefer it. So the fact that these look pretty happy and they're not sedimented is probably indicative that there's ongoing supply of whatever they're depending on, but it's not so much as to cause toxicity. And it's actually very interesting that sulfide is very toxic to a lot of organisms, as I said, but a lot of it, you know, it may not be here in that high concentration, given the number of organisms we see sort of, you know, sitting right next to the bacterial mats. But I'm not an expert on that part of it.
Yeah, whatever is associated with the mats certainly isn't bothering the eel pouts and the tube worms, the quill worms that we've seen.
Okay, so watch lead right here.
So I'm sure you know that some of the Pacific Northwest seeps were first found back and discussed maybe in 2015 or so based on the fact that fishermen were using these spots because they were very rich in marine life. And in fact, on the East Coast, on some seeps, we have found the same. The fishermen are often sitting there because they may not be aware that the seeps are there, but they're certainly aware that there's a lot of life in the water column that's feeding the species that they're trying to catch. And in fact, on the West Coast, they actually used the fishermen's observations to help locate where the seeps were.
Come in, video.
Yeah, and it's interesting as we learn more and more about these, their distributions and also their association with the different water depths. And Carolyn, most of the seeps that have been identified on our coastline here on the east coast of the U.S. are more in the shallower regions. Do you think just as we've discovered them more there, or do you think they're also as plentiful in these depths? Any ideas on that?
Okay, let's come up.
Yeah, I think they're mostly, at least, and I do think we've had good surveys of deeper water depths, but I think you're mostly going to find them at about 600 meters and shallower. And as we mentioned in the call this morning before we got on the public line, there's a huge number of sheep sitting very near the shelf break. So maybe, between 60 and 120 meters, particularly just upslope of here. And there are reasons that may be as well. The deep water systems are quite rare, and we used to think, down further south where you guys have been for the past few weeks, we used to think they were mostly connected to salt diapirs coming up from below and heating up the sediments and making something called methane hydrate, and those sediments break down and release methane. Of course, that does happen also in the Gulf of Mexico much more pervasively. But further north, we don't have diapirs beneath the deep seeps. Mm-hmm. And so that's why those deep seeps are particularly interesting, the ones that are 1,000 meters, 1,400 meters water depth. And they're relatively rare.
Huh. Interesting. Well, thank you. We're watching a tube anemone, excuse me, and it has several amphipods that don't seem to be too bothered by it. So maybe this is more of a mutualistic relationship. Maybe the amphipods are cleaning or eating off of the particulate matter. But it doesn't appear that the anemone is trying to actually eat them.
Okay, video whenever you're good. Watch lead, this is pilot.
Go ahead, watch lead. Go ahead, pilot.
So, I think they're gonna let the navigator know right now. We need to decide if we are going north, south, or west.
We've seen so many of these bacterial mats that we are modifying our track line here because we want to keep following them.
Okay, so I'm coming back to my port side.
Pretty stretched out, Fernando.
Copy.
So visibility is
pretty good.
Getting worse. It was really bad at the beginning, and as in the last
And we're having a pilot change. So we're going to hang out here for just another minute or two. And we've also been looking at our track line and looking at where the water anomaly plume data was, and it's just a little bit, slightly bit north of our track line, so we're just going to follow these a little bit longer, follow the bacterial mats. And we've also been looking at our track line and looking at where the water anomaly plume data was, and it's just a little bit, slightly bit north of our track line, so we're just going to follow these a little bit longer, follow the bacterial mats. And a little bit of background about these bacteria mats.
I got you loud and clear.
Okay, I'm gonna work with Nav here once they do the swap to get us a little closer.
The genus is Begiatoa.
This is
and they're named after an Italian medic and botanist who is named Begiato. I bet you in Italian that sounds a lot better.
Nice work, buddy.
But they're generally found in sulfur-rich environments.
Once you get settled in, let's try to get us a little closer to D2 so we can see what's going on.
And they are found on oxidized hydrogen sulfide. They use as an energy source.
Bearing 180.
Thanks.
Gonna come back into your view.
We were just talking about some of that.
Looks good.
Bridge, this is ROV Nav speaking. Yeah,
so we're coming
ready
south.
for a move.
I'm coming closer to you.
You are.
Right now I'm facing 193.
All right, range 30 meters.
So that's the oxidation of inorganic compounds.
Heading around a little further to the south. I'll center you up. Right now my camera's at 20
Actually,
degrees.
the oxidation of organic carbon,
a little
I think.
away. I'll let you know.
So anaerobic oxidation
scoot out,
of the methane is what produces the chain of reactions.
Once you get your scribbles down there. Did you talk to Watch Lead?
Okay, this is probably incorrect what I was looking at then.
Huh?
Follow the mats.
Follow the mats, okay.
But generally these are found in...
I think it's because the mats can exist in other environments, probably. Maybe Bob Carney knows, but I mean the environments we see them in here with seep.
So you
it's the organic carbon that's kind of in the base.
Stretched out, yeah, so southbound is just going to get us where he is. And then
everything that happens at these places.
Right.
Nice tracking.
And they're
based. Bob Carney just wrote in the chat room that oxidation of hydrogen sulfide yields a great amount of energy, so I assume that's what these guys are doing. Oh, I see what you're saying. The mats were using the hydrogen sulfide.
Yeah.
Michael later shown pilot.
Okay, I'm sorry. Yes.
Yeah, but different parts of the process. And so these...
For the listeners, that just goes to the fact that these processes that begin with organic carbon, the cascade of processes that feed what's going on here, they're very complicated processes.
Keep an eye
that
on these
make them very simple when we talk about them.
Right.
And follow the trail wherever it takes us. So you just keep poking along on those as we give you space, and
we're looking to head south.
Keep up. Right now we're headed south, yeah, but whatever the direction of the mats is.
All right, copy that. It looks like they're all over the place, to be honest.
Yeah. I guess maybe follow the bigger ones, or I don't know. Yeah.
Can't really see them in sonar, can we? I don't really have a good grasp of them here.
No. You can see the tendency, but this might just be that they're all over this area, and that was the track they happen to take. So it's hard to know.
Bacterial mat.
And so these bacterial mats can occur around cold seeps like we're seeing here, or sulfur spring sewage contaminated waters, mud layers in lakes, and near hydrothermal vents.
I see.
And the original species described is Begiatoa alba, but there was another species described just last year, leptiformis.
So is the altimeter in the back of the vehicle?
Yes.
Okay.
And it's the DVL in the back. There's bubbles came out right there, huh? Another set of bubbles.
You want to comment on that video?
I'm definitely moving it closer.
Is that full zoom, or can you come in a little more?
That's great. Look at those bubbles.
You got a little jellyfish hanging on your left rock box there.
Let's play with that a little too much. Said there's something coming out of the rock box?
No, there's just a jellyfish hanging out by your rock box. You can see it in your manip cam. You don't turn to look at it. Just you can glance at your manip cam just to see him hanging out. Sorry to distract you.
No worries.
Looks like you're looking.
Starting to catch up to you.
All right.
Getting in the configuration here.
Feels
Delta 10.
different.
Z-buys could be different than what you're used to or something. Is that what you're talking about? Thrusting is different? Or?
No, it just seemed like the camera was different. But what is like the normal just flying angle?
I like level, but
it's whatever, it's based entirely on what you're flying on. So on a really flat bottom, you tend to do a little lower than angle level. On a substrate like this, you might bring it more level or even a little up from level, so you can see what's coming and what's ahead a little bit. It depends.
Nav, this is watch one.
Go ahead, watch one.
Okay.
We're thinking that if we can, just get to the higher point instead of following lower on this ridge, it might be a good idea.
Understood. Yeah, right now we're headed south, kind of following these mats.
Okay, great.
And then we'll just kind of see where the mats take us, I think.
Yeah, and upslope might be more active bubbling, and we just saw some more bubbles, so good sign.
Understood.
Do you want to trace the bubbles?
Go ahead, Bridge.
No, that's okay. I think we'll see more bubbles as we move.
Understood. Yeah, you can change the heading a little bit now. 10 degrees I.
Okay, I'm at Delta 10. Camera's at 45.
Copy that. I'll push up.
That looks like, I think you're okay. This is good visibility for you. You can see yourself at the bottom a little bit. Trying to be a little close to you.
Understood. Making a heading change, pilots, but they can stop it, make a move if we need, and show us the position.
Roger.
So yeah, science wants to go uphill.
Uphill,
follow the mats.
Okay.
So I think that's what we're
follow the mats or uphill. Cause
are they not the same direction right now?
They're pretty much the same direction.
Okay.
Let me aim uphill. Just take a peek here. Pilot, my head's rocking right. You're fine where you are. Just keep doing your thing. Yeah, maybe like 200s uphill from my sonar. So we can do the rest around that. I'm at 185, lined up on you, pilot.
Yeah, Hypex got uphill like 213.
215, maybe 3. Okay. Yeah, so we'll get moseyed over towards 200. I think if we do short, like 10 or 20 meters
a couple
at
of black
decimal
belly
1,
rosefish here.
we can like stay right here with them, and so you can see.
A slight depression.
Yeah, we do that.
And
So.
Looks like we're running out of the mats a little bit.
Yeah, 200.
So should we follow the mats or should we go to 200?
I would say follow the mats, actually.
Where are the mats?
There's some over here.
Yeah, that's still 200.
So I feel like I keep... I guess what is the Z-bias? Is that just how...
That's how hard
is that controlling the buoyancy, correct?
It's how hard it's thrusting down without you touching the stick.
Okay.
So it's sort of like a trim on a helicopter, if you will.
Gotcha.
You know, like sometimes on a helicopter, hobby helicopter, if you let go, it might like drift one way. So you can give a command slightly to the right to sort of keep it. This would drift up, so we give a command down to keep it down.
And we got a question from the audience, from Shireen Gonzaga. She asked, did we get the worm in the biology sample 1 from yesterday? And the answer was yes, we did get the worm. And we
like to my,
did look at it under a microscope.
I'm coming over to
and
the
we
left.
had
I'm
an
right
ID
yet, so
that came from somebody
you can go over to where they are.
in Twitter.
Have you seen this thing yet?
And I can look
the
that
fish
up.
that isn't moving.
What else did we find in the sediment? We found
Grab the green eye.
another type of worm, and I'm not sure what that one is. We did not find any brittle stars. We really didn't see.
They're right there.
Yeah.
We had another sample had some sediment in it as well, and we didn't see any brittle stars in that either.
In the back of your mind.
And so we looked through the sediment,
Could do 10
and
meters, 200. Copy.
we sieved some and
come up for a second?
to get the larger particles and looked through that, and I think that's where some of those other worms came from, besides the larger ribbon worm that was the target of the sample. And when Leslie returns from lunch, we can have her talk about what else she saw looking at the sediments under the microscope.
Good copy. 130I.
Okay, pilot, we're coming your way a little bit.
Copy that.
Following the mats. Do you see? Is there one off?
Yeah, there's one right directly ahead of me. Further on, it looks like.
Connect the dots.
Keep the trail going if you can there with the targets.
All right.
So the one
I
that
feel like the vehicle is high, but my left toe is very close.
Yeah, so that's because right now, if you look at your sonar, the slope is to your left.
Gotcha.
So if you were to wag and look at the slope, then you wouldn't feel so high.
Would it be better to face the slope and move kind of parallel to it, rather than trying to do it the way I'm doing it?
It would.
I mean, you're not going to see as much over there, but
you're going to want to do both. So you're going to want to see where you're going, but you're going to want to keep what's interesting in view at the same time.
Alright.
So you're going to want to look out, make sure there's no lines, make sure there's no things that you're going to run over.
So if I face like more like this, I can drive that way, but
yeah, you can drive any direction you want, whatever's.
Yeah, you're doing a good job.
Yeah,
Lars.
I mean, I just feel like I feel like I'm too high to be able to see anything decent, but at the same time, if I go lower, I'm going to be dragging that toe through the mud.
It's the game we play, sort of sidling like this.
Yeah.
Go ahead, watch lead. No. He has you latched on. Yeah, we can have him undo that.
What was this?
See how the wash 1 and 2 are latched on?
Yep.
Just to undo that, they'll just listen to you when they want to hear, or if you want to make sure they can hear you.
Copy that.